So before that, students need to know some stuff that is not in first year, not in first year anything, basically. You know, what is a probability distribution? Where are they going to get that in their first year courses? So we have to teach them that. What's the expectation of a random variable? Okay, very valuable stuff for them, but they're pretty innocent, most of them, about this. Even the ones who have taken a course on biostatistics need to have it taken from a practical point of view. We're going to be seeing a particular probability distribution. Okay, it's one that comes up all the time in biophysics. They're going to need some really rather primitive coding skills, which we get pretty early in that class, and not much else. Okay, they're going to need to know what's a semi-log plot, but you know, not a whole lot of background here, though there are these things that are not in first year physics. Now, I think it's really important if you want students to work to give them a puzzle, give them something to ignite their scientific curiosity if they have any. So here it comes. What we want is a story that begins with an earthquake and works its way up to a climax. But yeah, that's Samuel Goldman. And here's the question. How can pulling two things apart strengthen their bond? It's crazy, right? It's ridiculous. So before we get into asking how it can happen, we should ask, does it actually happen? And what might it be important for? Well, you know, everybody on the planet is extremely vitally important in this picture right now. Right? It's a T cell coming up against another cell and trying to decide whether it's infected with COVID or not. Okay, and how does it do that? Well, you know, there's some receptors, and the receptors have to engage some MHC complexes. And they don't just bind, they bind and then they start pulling. Okay, and that's mechanobiology, isn't it? That's why everybody on the planet is interested in mechanobiology right now. Okay, there's a crucial step involving T cell recognition, T cell activation that involves pulling with a force, like an Isaac Newton kind of force. And that T cell is trying to decide how strongly is it engaged with that MHC by pulling on it and seeing how long the bond lasts. Okay, now you, the student, might be engaged. Okay, this is important stuff. So I want to say, well, cells communicate biomechanical forces. Cells form bonds with their neighbors, then actively test the strength of that bond. Some bonds, surprisingly, get stronger under stress. It's cat bonding. It's surprising, but it can be understood and modeled, and that's mechanobiology, and that's why we want to study it. Okay, and there's some other stuff, and listen, I don't think I have time to go through philosophy like this, so, but the last point, when you make a scientific animation all by yourself, you, the student, you never forget that first time, and that's what I want to give them. Okay, so before we get into all that theory stuff, does it really happen? Well, we live in an era where it's possible to manipulate molecules one at a time. And here's a pretty recent example. You know, optical trap, there's your T cell receptor, there's your major histocompatibility complex. No living cells anywhere. You just bring the two in apposition and you pull on them and you see what happens when you pull on them. And well, you know, you get timed courses like this. That's the kind of experimental data we now have available to us. And over on the side, you see that there's some bond lifetime that maxes out at a non-zero pulling force. And it's exquisitely sensitive. One little amino acid mistake in the peptide makes a huge difference in the lifetime of this bond, and that's what the T cell is trying to look for. So how are we going to model that? Well, everything just comes out of this one little line of code. You know, simulate a Bernoulli trial. Pull a random number and see whether it's bigger or less than 49%, and that'll be a Bernoulli trial. Okay, we just have to embed that in a little bit of code, but it really doesn't take a lot of code for students to write a little simulation of Brownian motion with drift. That's why it's 49%, not 50%. And you see a great deal of moving around, but of course it's better with a video. So it's better yet if the video that students make for themselves. So here's that guy moving around in that trap, and though he's in a potential well that forces him to drift off to the right, you know, he's taking his time getting there before he finally gets there to the right. And you learn all sorts of things from watching that movie. Okay, that random walker, he's not trying to get out of the trap. He doesn't even know there is a way out of the trap. He just blunders towards it. He's not creeping up towards the exit. Eventually just stumbles on the exit, wasting a lot of time. Okay, there's a lot of physics with biological context there just by watching ground in motion, which you get by that one line of simulation code. Okay, then we can move on to motion in a trap. Suppose the force isn't a constant, but depends on position. There's a potential energy well down there. And here's what you get. And it spends most of its time in the bottom of the trap, but once in a while makes big excursions. That's the sort of thing we're interested in if we're interested in the formation and breakdown of bonds. So that's a fun movie to watch. And well, once again, spends a great deal of time trapped before it finally makes it to one of those exits. Is it boring? No, it's incredibly exciting. Is he going to make it? Is he ever going to make it? Looks like he's never going to make it. But then, with no warning, oh, he gets out. So students can make that themselves, and then they can watch it and learn things from it. Then they can take the bigger step and say, well, what fraction of time does he spend at various places? And they discover the Boltzmann distribution just from that stupid little simulation, which didn't seem to have the Boltzmann distribution in it, but it comes out. Okay, I think that's much more concrete than the way we teach it in Stat Mech course. You can discover that the distribution of escape times is exponential in form, depends on how high the barrier is. Oh, wouldn't it be better to watch a movie? Okay, well, that went by kind of fast. Let's stop that. Here's the probability distribution function of a whole lot of random walkers all released at the center. And oh, almost immediately, they equilibrate. And then the probability of escaping just goes up linearly in time. And there's your constant probability per unit time of escape, okay, given to you in a concrete form. How long does it take? Well, it depends on how high the barrier is. You discover the Arrhenius law, okay? What about those catch bonds? Well, there's Velcro, and Velcro works because there's two escape routes. There's the difficult escape route, and there's the easy escape route that you have to push in the wrong direction. And we can model that, and we can find that the lifetime initially increases with applied force, and then with a very big applied force, it starts decreasing again. Then we can say, yeah, but is any of that really true? Does any of that really apply? Well, this is not a single molecule experiment. This is a single cell experiment. This is the activation of T cells. This is what really matters for immune response. Does the T cell turn on or not as a bead is brought up against the cell and pulled? And once again, low pulling force doesn't activate, needs some pulling force to activate that. That cell is engaging, bonding, and then testing the bond by pulling to decide whether it should activate or not. And how interesting and totally unexpected when people did these experiments, tangential force is different from normal force. And oh, how interesting, when cells crawl across each other, they generate a tangential force and not a normal force. So suddenly we're in the 21st century talking about stuff that's in the headlines, but viewing it through a simulation that was very straightforward for them to do for themselves. Do I think they can really do it? Yeah, every year they do simulations and other coding challenges that are equally complex as this one. It's not very complex. So, okay, I should wrap up. Some of these ideas are also beautiful, by the way. Now we can say, well, what does that mean? I think a beautiful physical idea is something that's surprising, but in retrospect, inevitable. Simple, but unexpectedly general. I think the biased random walk has all those properties that we want students to know about. We've also seen how you can get a concrete discovery-based approach to things that are normally presented abstractly. It's amazing how a handful of physical concepts can be used to understand all these problems at various levels. That's what I like to teach as a physicist, but I love to teach it in the context of biophysics because it's obviously important, unlike, say, Atwood's machine. Okay. Things I gave you today are all available. They're in a chapter that I put on, that Annie put on her spreadsheet. And movies I showed you, I'll put them in the chat so you can watch them yourself. It's all going to be part of the book, and I'm going to thank you for your attention and take all the questions. If any.
Annie, how do you want us to ask questions?
You can just unmute yourself and start asking. I think that would be fine.
Bill, that was a really beautiful lecture. I never thought of showing of showing something as simple as Boltzmann distribution through by showing this kind of inevitable, simple, but it's surprising but inevitable. And what do you do to teach the other part of what you showed at the very end? How do you convince how do you convince students that these principles are very general, simple yet general? So you showed the surprising yet inevitable really beautifully. No one can leave that lecture and consider stat mech to be anything other than surprising yet inevitable. What examples do you like to use to show generality?
Okay, well, throughout the semester, we try one example after another. And I think the idea that everything is jiggling around thermally is something that's not in freshman physics anymore. Like 50 years ago, that was in freshman physics, and now it's not. So it's a big surprise to them, and we have to show it to them in all sorts of contexts. And by the end of the semester, once you've done 7 or 8 case studies like that, they're starting to see it. I don't have a lot of specifically mechanobiological examples yet. They quickly get kind of involved, and I wanted to choose one that I thought people could get their hands on already in the middle of the semester and not wait for the end of the semester. But there are plenty of other examples of thermal motion leading to surprising physics. For instance, involving electrophoresis and other laboratory techniques that are important to people.
Thank you.
Do right now.
Hi, can I ask a question?
Yeah.
By the way, I love the examples that you covered. Beautiful. I used to teach a biophysics class when I was a faculty member at a liberal arts college in Louisiana. And if I were still there, I would have copied and pasted exactly what you said into my class there. It's fantastic. One question that I have for you is what your experience is with the kind of students that you have dealing with probability distributions. This is something that I always found that is challenging, even for students who have had one semester of physics, 1 year of physics, an introductory course in physics, 1 year long, calculus based. Even though the course covered probabilities and in labs, probability distributions and how standard deviation of the means, they've had that in math, they always struggled with probability distributions somehow. What is your experience with your students?
Yeah, that's such an important point. Like, even the students who took a whole semester of biostatistics have no idea how to go from a situation to the distribution that might be describing that situation. So we have to spend a third of the semester, or at least a quarter of the semester, on exactly that. What is a distribution? What is a continuous density function? How do density functions transform? Because you can't pick up any piece of literature if you don't have that backbone of understanding. So we just have to make room for it because they're all quite innocent about that. I'll just say one thing though. So many things in probability are presented to students in this totally abstract way because the pedagogy got frozen back at a time before people had their own computers. Okay, so many things are so much more real if you just write 5 lines of code and see it coming out yourself and make a picture. So in the first edition of that book, the one that's actually available, about a third of the book is that sort of thing. You know, what is a power law distribution? They think everything is a Gaussian, but that's not true. You know, what is a long tail? And so the more they can do themselves or in teams using open source software, the better they'll understand it. Like, you know, the central limit theorem. Every time I read the proof of the central limit theorem, 15 minutes later, I've forgotten how it works. Okay, if you just add 5 random variables together and make a histogram, oh, that looks like a bell curve. You know, then you remember. So that's my point about teaching, is that you now have open source resources such as Python for students to do industrial strength computation themselves on their own. You can dedicate a few classes to that and then let them learn more as the semester progresses.
Okay, yeah, thank you.
That sort of segues nicely into my question for you, Phil, which is, when are students actually coding live during the lecture while you're doing this? Or do you integrate that with separate discussion sections or?
Okay. So what I've done in the past has been to have 2 in-person lab sessions right at the very beginning of the class. You know, lectures 2 and 3 are not lectures, they are lab sessions where I tell them a little thing and then we wander around the room with the TA and me, you know, troubleshooting and that sort of thing. And then after 2 such sessions, then we just feed them a new skill every week. You know, this week we're going to need a loop. This week we're going to, you know, okay, but we don't let it dominate the class after the first 2 sessions. Now, how we're going to do that remotely will be an interesting question, but you know, we'll try to simulate that in some way. I did write a little boot camp book with just enough Python to do the problem sets in a biophysics class. You know, a really, really short book with, you know, solve an ODE, some recipes like that. And that's helpful to them as a backstop as well.
So so I guess sort of the other question that I had was, do you expect that they're writing the code and practicing, you know, running a Brownian trap and things like this while they're actually engaged with you in the lecture material? So do they have their computers with them?
No.
Okay.
After those first sessions, they do that sort of thing in homework. But I assign them partners, and I assign teams where one person has self-reported as having high skills and the other has self-reported as having low skills. And I say to them, you know, the one of you who has the more background is not allowed to touch the computer, but can only give advice while the other person touches the computer. That's how we do the first couple of sessions. But after that, no, I am going to have breakout sessions where they talk about things, but it can be very time consuming to, you know, figure out how to import that data file and stuff like that. That could eat up the whole hour, so we're not going to attempt that.
All right, great. Thanks, Phil.
Oh, can I quickly, sorry, Annie, can I
Sure.
quickly ask a question?
Mhm.
I wanted to wait for other people to finish questions. So Phil, I was really curious, you know, you showed, you were talking about the type of students you have. I'm curious what your experience has been with the biology students. You had a bunch of biochemists in the class, you know, because this is something that I've thought about doing analogous things in my cell biology class, you know, dedicate a class or 2 on some of these topics, because I do cover T cell activation. I do cover a bunch of cytoskeleton stuff, you know, where this obvious mechanobiology that can be simulated in very simple manners. So how do you, what's been your experience with, you know, my students are typically juniors and seniors, but there's actually a more growing number of sophomores who do just as well in the class currently, you know, but I'm just wondering, they've not typically taken intro physics by the time they take my class.
Okay, yeah, that's a very different kettle of fish. You know, by setting the bar at freshman physics, I only get the tail on the distribution of biology majors, the math-friendly distribution tail. I do believe that we must do more and better for those students. Certainly everything I said, you can show them the movies, but I don't think it would be too difficult to at least get the first of those simulations. You know, the Brownian motion with drift is already very exciting. And I don't think that would be too difficult. There are some Python resources like VPython that make the burden of coding much lighter. So yeah, I didn't show you that movie, but I have one that's literally 4 lines long and does something that looks real. It's hopping between two states in a trap. Then you show them the video micrograph of a bead hopping between 2 optical traps, and it looks similar.
Mm-hmm. Right. Yeah, thanks. Yeah, I like that idea of just, you know, not using simple exercises and using movies, which I do, and they really enjoy having movies embedded in lectures and stuff. Sort of breaks it up and makes it more real and dynamic for them.
Yeah, exactly. There are things you will never see in a still image that you see with a different part of your brain in a movie.
Thanks.
This is perhaps a weird, like not helpful comment for this discussion because it's a wider curriculum issue. But at UCLA, they've been developing a mathematical biology course for biologists that involves teaching them like coding skills and running through, you know, all kinds of interesting, you know, and or gate programming and things like this, transcriptional networks. So using it for biology, it's a useful way, I think, to teach some basic coding skills, but also preps them for upper div classes where they might do things like Ram's suggesting. But I know that's a huge thing. I'm not sure all universities are starting to do that yet, but something for people to consider also, an integration of basic coding and quantitative mathematical biology for your biologists at lower division. So this is in the first year they do this. Could be something that could be on your mechanobiology agenda, but probably a much larger problem.
Well, now that the barn door is open to things that are not strictly mechanobiology, feedback control is tremendously important throughout all of biology. And, you know, making a phase portrait that shows you in a visceral way why that feedback control is going to give you a toggle or an oscillator or whatever. That's about the other half of that book I wrote called Physical Models, is just getting students to do that for themselves.
So I thought I'd just piggyback really quickly on that comment about getting programming skills into the lower divisions. One thing I'm trying to do with Westchester is I'm trying to work with all of the faculty to replace all of their labs. It doesn't matter which class, to replace all of the labs with doing their plots through R or MATLAB or Python. That way they're just seeing it constantly. And, you know, if they're going to use Excel to make a plot, why not teach them from the very beginning some basic coding skills? That way, when we get to these simulations, that we can just bypass a lot of that because they're pretty much used to it already. I don't know how long it's going to take for this to be implemented at Westchester, but it's one of my sort of long range goals that I think would be helpful for our students.
Yeah, that's terrific. You know, the earlier they start doing that, the more likely they are to just naturally turn to it on a future occasion. Can I just add, you know, it's pathetic, but the skill of being able to grab a data set off the web and wrangle it into a form that your software can read, it's really important. You know, we spend a lot of our time doing that sort of thing. That should be inculcated too. You know, and where did my browser put that file, and why can't my Python find that file? You know, all that stuff. You have to do it 3 or 4 times before you stop being afraid of it. And that's something I also want to incorporate into even that very early week. Just get a data set, get it into your software, make a graph.
Great, thank you. I hate to interrupt this conversation, but let's hear from Ram and Becky, and then maybe we can come back to this with their contributions as well.
Okay, thanks everyone.
Thanks.
I just want to tell you a little bit about what Ram and I want to talk to you about. And first of all, just welcome everyone and just tell you how this particular roundtable got started. This grows out of a meeting that Annie and I had with Adam at Bryn Mawr actually it was at Penn, but Adam from Bryn Mawr a few months ago. And he had this great idea actually of bringing together people, professors who he was thinking of primarily undergraduate institutions, but that's not necessary, to talk about how actually they are teaching mechanobiology and incorporating mechanobiology into their curriculum and their courses. And we had this idea that this was something that would become self-perpetuating run by the educators who are particularly involved. And we hope that that will actually happen. We're giving you some introductions today, but we appreciate that we're talking to people who are extraordinarily experienced at teaching. And so Ram and I are going to give you some examples about mechanobiology classes that we teach for graduate students, but we're very interested in hearing your suggestions and tips, because I think that'll be useful for the whole group. Can you give me the next slide, Annie, please? So there are 3 courses that between the two of us that we teach at the graduate level that have mechanobiology content. So the first is a large multi-site lecture class. Some of you have actually had students take this class. It's open to anyone in our network. It's always been done online, called the Integrated Mechanobiology of Plants and Animals. This coming year will be our 3rd year giving this course. Obviously, it's undergone a lot of change in the last couple years because it is a new course and is a little bit of an unusual topic, but we'll give you some ideas of what we've learned. We've been very, very aggressive about seeking input from students, input and suggestions. So much of what we've changed is actually based on student suggestion. The second class is one that I've been involved in for, this is an every other year class that I've done myself 5 times. Ram has actually come and been one of our guest preceptors at one point. This is a journal club format. A small class of students, generally 8 to 12 students, we cap it. It's called the ECM, Adhesion Receptor Signaling and Translational Biomechanics, and we offer this at Penn. And then the third kind of class is one that I've been involved in along with a number of other CEMB faculty at Penn. It's a cell signaling class called Advanced Topics in Cell Biology and Physiology that runs for a full year and has in the last 3 years added on a 1-month tissue mechanics module. This class has about, I would say 15 or 16 students. And this 1-month module features 1 week each, namely 2 classes from various specific professors in mechanobiology. So could you go on to the next slide, please, Annie? So one thing that is common to all of these classes and that has been a real focus of what we've done is helping students integrating information and concepts that span the physical and biological sciences, i.e. mechanics and biology. And this is particularly an issue at the graduate student level because the students come in with very, very different backgrounds. Unlike the students that Phil was talking to you about, who I don't want to say they're homogeneous, but you can catch them early. The students that we are seeing have already gotten into very specific grooves. The biology students, actually, can you go on to the next slide, Annie? The biology students in many cases are very scared of math, very hesitant to incorporate math into what they do. I think some of Phil's suggestions are actually very good, might be good for many of our classes. Surprisingly, the physics and the physical science students are actually also have problems with this kind of curriculum. Many of them are really blown away by the amount of information provided in biology lectures and by the I don't want to say amount of memorization, but by the acronyms. It's turned out to be a huge problem. It sounds trivial, but that's one of the challenges that we've seen in all of these courses, is that when you're trying to have a class that involves graduate students, they do come in with these backgrounds that you have to deal with. The second problem that we, or second challenge that we've encountered is that faculty expertise may also not be as broad as the course demands. You know, our goal is really to sort of develop some mechanobiologists, but we don't necessarily have them at the level of the faculty. We all have areas of expertise that may be stronger in one area than another. And then the third challenge that we found is that certainly for every class that you teach, you want students to be able to integrate and keep up. We have found that this particular topic that is even, if I can say, even more true than in other classes. It's very important that students keep up. They can get lost very, very quickly, particularly given their heterogeneous backgrounds. And that really it's not going to be a valuable class for them unless they're able to integrate what they're learning. So if we can go on to the next slide. So Ram is going to tell you about this, our large multi-site lecture class, the Integrated Mechanobiology of Plants and Animals. And I just want to add that we are happy to provide our lecture recordings, our syllabi for any of these courses, the papers that we have considered. Any of that would be available to any of you. Please just contact me or Ram. You know, today we sort of designed this talk to be a little bit more general about the general challenges, but we do have some specific resources if you'd like them. So I'm going to let Ram talk about this class, and then I'm going to talk about the other two classes a little bit at the end.
Okay, thanks, Becky. So as Becky mentioned, this multi-site lecture class is a large class format, and you know, from the very get-go, the challenges that we knew we would face was that this would be a diversity of students and also from multiple locations, right? So as Becky mentioned, this class has been sort of essentially has had an online version of flavor to it from the very get-go to accommodate that multi-site requirement. One of the things that we wanted to touch on is, you know, just in terms of all of you are thinking about incorporating mechanobiology from different perspectives and at a different sort of courses. And so, you know, while this course we are using as an example, and, you know, Becky is going to talk about the other two in just a bit, you know, this entire course is really focusing on mechanobiology. Now, you may be interested in a module, as Phil mentioned, or some blend of this. And so, you know, it is important to define your learning objectives. And for this course, since this is the required course for our CEMB graduate students, you know, we really want them to learn the fundamentals of mechanics and how they apply across both the plant and the animal kingdoms, and also spanning scales. So there's a lot of topics that you know, we throw at them that we want them to grapple with, and so that raises a challenge that, you know, that ties into the integration topic that I'll get onto the next slide. But through the center, you know, one of the advantages of the center that we capitalize on is the breadth of expertise that we have among our faculty. And so this course has been a team taught class. So we have sort of a variety of topics, some of which are introductory, where we provide some basic chemistry, some basic mechanics, some basic cell biology to sort of orient the students, all students really, whether they are have a training or background in physical sciences or the biological sciences. Even the students in biology have a wide range of experience. So we give them sort of a foundational information from the very get-go, and then sort of build on that and get into more complex and more integrative sort of what we call big picture type things that connect two or more of these concepts or topics towards the end of the class. So having this incredible expertise among our faculty has been really helpful to tap into, you know, where people can give those lectures with that specific expertise that they're bringing in for the topics that we want to cover in the lecture. The lectures are recorded and they are available to the students, and this has been really helpful because, again, the students have lots of different backgrounds, and so it's really helpful to them to go back and, you know, watch a portion of a lecture or a whole lecture or whatever it is as a review and as a resource for them that they can have for the entire semester. We have a strong TA support, and so the TAs are really critical to help students along. You know, so students who are struggling with the, particular topics or concepts can, of course, reach out to the instructors. but also the TAs are sort of more of a near peer type of help situation that they can take advantage of through meeting with the TAs and working through problem sets, et cetera, with the TAs. We've been, one of the sort of things that we have sort of noticed is with student group projects, especially if you put together students that have, some who have a strong background in a particular topic and the other in weak. As Phil mentioned, that's really works great, but I think Phil emphasized the key point, which is that you need to then make sure that the students who are really good at it don't just do all the work, which is what we found to be a cautionary note with these group projects. And so you do need to have mechanisms in place to make sure that they are learning from each other, and that the person who is more adept at a particular skill or topic is sort of facilitating the learning for the other student and not just doing the work for them. Can we move to the next slide, please, Annie? And Becky, please add anything if you had some points that I'm missing here. So integration is really the key. I mean, the center is all about integrating plant and animal mechanobiology across multiple length and time scales. And we want to capture that in our course. And so what do we mean by integration in the course? What we mean is to help really students map out explicitly the connections between the different lectures, because otherwise it can just come across as a smorgasbord of a variety of topics, and students really struggle at understanding, how does the basic biochemistry connect to Brownian motion or mechanics, what's happening at different scales, between plants and animals. And so we have sort of played around with a variety of ways to do that. And in fact, we continue to add to how we do integration in the course. So one of the things that we have done is all of our lecturers provide food for thought kinds of questions. So these are sort of, open-ended sort of inquiry type questions that we give to the students with this, before class. And they really sort of provide a mechanism for students to think about ahead of learning the material, about certain topics and think through them on their own. And so starting this year, what we're going to do is we're going to pose these questions to them. They will be provided these questions before each of the lectures, and they will have to post their comments on a Google site by the previous night, and they will be graded, right? So the grading will be based on their participation. Did they give a real legitimate shot at it? It's not about right or wrong answer, but it's really making them think about it and sort of think, for themselves as to what they think might be intuitively might be happening in a particular example, for example.
Can I just add
Yeah,
one thing?
sure.
This is something that we're doing. You'll see this in all of our classes. This is something that I learned from another faculty member in another course. It has worked extraordinarily well for us. It's a way for us to, the students have to keep up, and they can't just do it in the 5 minutes before class. They can't do it at 2 in the morning. They really have to be organized about their reading and thinking. I think it's made them enabled them to be much, much more integrative and to sort of stay up to speed much better. It's also a way, because you as the faculty member can read through these the night before, it becomes immediately clear who's having problems of various kinds and who is not. And they also come up with some very interesting ideas. So I should say Guy Jenin, who participates in this class, who is actually the site director at WashU, has been a big proponent of these food for thought discussions. And the students have actually requested more of them. So this year we've added on many more.
Sorry, Becky, could you just summarize briefly again how it works?
Yeah, so we give them on the day of a lecture, we have the lecturer has provided 1, 2, 3 food for thought questions that are designed to integrate the material from that particular lecture with the whole concept of mechanobiology. Ram and Guy and I may adjust them a little bit to bring in some concepts from previous lectures. And then the students are asked to provide written responses. They don't have to be long in any way, you know, sort of on the order of a paragraph, onto a Canvas site or some kind of site that is public for the class by 9 o'clock the night before. So, and they have to meet that deadline. They don't have to get the questions right, but they have to show some evidence that they've actually thought about it. And so they're graded on that. I think we're pretty relaxed about that, to be honest with you, but the goal is that we have students participating, you know, sort of throughout. And these questions are designed to be really sort of open-ended and integrative. And then the next day, the first 15 or 20 minutes of the class are a discussion of those questions before we then go on to the next lecture. So something new that we tried last year, we're expanding it this year, but it's worked incredibly well. But I would say the idea of putting comments on a journal club paper or anything else by 9 or 10 o'clock the night before has been extraordinarily successful for us.
Great, thanks.
Yeah, and I would say that in my own undergraduate cell biology class, we do something similar to this. And actually the students really like it. They seem reluctant the first couple of times, but then once they see the value of it and how it helps them really make those connections for themselves, then they really like it. And that's something that I see again and again in our evaluations, where they say that that was something that they truly got a lot out of. And so I think, you know, as Becky mentioned, Guy has really been a sort of a leader and a proponent in this area. And so we're sort of formalizing it a lot more this year, including the really dedicating the first 15, 20 minutes of every lecture for these discussions, you know, where we have discussions about these food for thought, based on these food for thought questions that tie into material that was covered previously and that also anticipates material that will be covered in subsequent lectures.
For just your context, how big is this class?
This is about 40 students,
40, 50,
I think,
yeah.
but spread across Penn, WashU. We've had students from UC Merced, from Bryn Mawr, from NJIT. It's open to any student in our network. It's an independent study grading mechanism at individual institutions. So you have to have a specific a faculty member at that institution who will take responsibility for the grading. Although we actually do the grading and recommend a grade, but it is formally given by the site professor, site director.
Okay, I'll pick up the pace a little bit just in the interest of time. I think I've already mentioned some of this. So the way we have organized the course in the past, but particularly more so this year, is sort of have 4 or 5 introductory type of lectures, as I mentioned, to cover the basics of sort of basic biochemistry, cell biology, mechanics. And then we go into a couple of modules that are a bit more specific, where we drill it down into cytoskeleton, extracellular matrix, cell wall, statistical mechanics, and so on and so forth. And then we really sort of wrap it up with what we call a big pictures kind of module that ties together these concepts related to, you know, how information in biology links across scales or between kingdoms and so forth, as a way to, again, sort of really get to thinking in an integrative manner in this course. We have a few journal clubs sort of scattered during the semester. And again, here we sort of judiciously pick papers that sort of work well to highlight connections between the topics that we are covering in the course. And the course culminates in this a final project that the students do. It's sort of a mock grant application, specific aims, paid style, that needs to incorporate a concept that, again, is integrated directly across scales or across kingdoms, and brings together couple or 2 or more of these kinds of things that we have covered in the course. And the students have help with this. You know, many of them are doing this for the first time, or even if they have done it before in some undergraduate courses, it's still tough. And so we help them through that, you know, in terms of coaching them through the steps to how you develop one of these what is a good question that is an approachable question? How would you attack that problem and so forth? And so the TAs and the instructors work with the students for that. Can we get to the next slide, please? Becky is going to talk about this.
So I'm just going to talk briefly because I know we're running late. The second course that I wanted to talk about is this 8 to 12 student journal club format small class. This class, which we've given at least 5 times, is actually, I would say, works extremely well. It's given by right now by me and by Rob Mock, who's a bioengineer. So we know the students' backgrounds in advance. We do have a mix of pure biology students as well as bioengineers and chemical engineers, and we try to sort of straddle that. One of the ways that we do that is we give introductory lectures at the beginning of the class and in the middle of the class. I would say a total of about 8 introductory lectures that can include sort of basic biomechanics as well as more sort of advanced biomechanics. These are short lectures, 20 to 30 minutes. And are relatively straightforward. We also provide supplemental resources. So the goal is that the students may need to do a little bit of work on their own to get themselves up to speed, but we try to make it as easy as possible for them. This is a class that's based on journal papers, and so we have small group or single student presentations for each paper. Students can choose who they want to present with, and it is a mix. To be honest with you, though, we don't tend to have, you know, students, a biology student pairing with a bioengineering or a chemical engineering student. We tend to have students who know each other. And so they do sort of have you know, the group will have a weakness that they're going to have to tackle by themselves. So they, it tends to work out well in this particular case for group presentations. Again, they have to post comments on the paper by 9 o'clock the night before. We've seen that makes a huge difference in how well prepared the students are for the discussion the next day. And then the one other thing that we added early on in this course was that 3 times during the semester, we have small groups. Those are usually 3 to 4 students, lead what we call integration sessions. And their mission is to integrate everything that they've heard up until that point. They can ignore certain papers if they're not so interested in them, but we really want them to sort of try to integrate the concepts, tell us what we missed. I would say this past year, the integration sessions have been the absolute best. And I think that's because we've required these comments to be posted the night before. The students are really much more on top of every single paper. So the students have actually been telling us, you know, well, you know, we went through this concept, and this concept. We can see that more research is required for this area and that area, but we really think that Becky and Rob should have given us a paper on this topic. So it really has, the students have really taken this, this concept to heart. And they're really sitting and thinking about what they've learned and sort of trying to integrate it into into sort of the big concepts of mechanobiology. If anyone has any questions about that, I'm happy to discuss it in more detail because it's turned out to be much more than just a summary. They really have managed to integrate what they've learned, but it's taken some hard work and some time on the students' part. The next slide, please. So the final format that I wanted to talk about is when you just have a small amount of time to add some mechanobiology to a class that may or may not, that is not primarily mechanobiology focused. And this is, as an example, I've been involved in a cell signaling class. It's called Advanced Topics in Cell Biology and Physiology. This is purely cell biology students. So I would say this kind of class may bring you a much more heterogeneous group of students. It's been very important for me to know what the expertise of these students is and to adjust appropriately. One format that has worked for this kind of class is that each week is started with an introductory lecture that brings the students up to speed And then it's followed later in the week by a student-led journal article discussion. So the intro lecture, which the faculty member would give, is really designed to help them understand a very specific journal article. And then the students, a small group of students, will lead that journal article discussion. Again, students are in the course are required to put comments online a day in advance, but these actually, the deadline is a little bit sooner, and the students who are giving the presentation will incorporate these comments into their presentation, and that has worked very well. So for this particular module, we've had 4 different mechanobiology faculty involved to give the module, and overall it has, this particular format has worked very well, sort of plopping, you know, a week at a time, a specific concept into an existing course that, that really isn't mechanobiology oriented. So next slide, I think that might be the last slide. Yeah, so that's the end of what Ram and I had to say. We are, as I said, we're happy to answer questions or to provide specific resources for any of these courses.